The title of this presentation is Assessing the Efficacy of mRNA Vaccine Candidates with in-silico Workflows. To begin with a presentation overview, we'll be describing and drawing upon some previous background that's presented in another presentation in the channel, which we're going to be able to interpret and repackage in a slightly different way to demonstrate how we can make use of deep learning and machine learning algorithms for structure prediction and enhancing our knowledge of different biological processes that can have impact on the genome, transcriptome, or other biological entities. And to do this, we'll be introducing a mathematical workflow which is rooted in deep learning for studying the activity of COVID inhibitors. And this will allow us to motivate looking at the presence of protease inhibitors and how each inhibitor interacts with binding on the surface of the COVID-19 crystal structure, and to be able to make use of our algorithms and to look at some ways in which different conformations could be produced from an open source workflow, which gives us an encouraging starting point that we could then use to further refine on and build our intuition on with proprietary neural networks or proprietary algorithms. We're going to refer to the protein data bank because it has a PDB structure which encodes coordinates for the structure that we would like to study the surface and dynamics on from different other molecules or ligands. And this will allow us to establish points of comparison between the predicted binding affinities of the deep learning framework and other source molecular docking frameworks. So we first need to start by looking about how we can obtain the PDB structure from the protein data bank. And in this case, I provide some information for one of the structures. And the thing that's so great about the protein data bank is that it provides some biological type of assembly of different types of conformations from ones whose crystal structures have been recorded publicly. And it allows it to be downloaded in a variety of formats so that we can make use of different types of formats depending upon which type of input data could maybe be easiest to feed into a machine learning or deep learning algorithm. In this case, it's demonstrating to us that we can look at different types of properties which would indicate likely conformations or likely modes of interaction between different type of compounds. And then looking at how these compounds would interact is important, because if we think about different scales of interaction, there can either exist macromolecules which are interacting with one another, which are each approximately of the same size or dimension, or you can have something which is a bit simpler, in which maybe one compound is a lot smaller than another, and then we would be more in the case of having a ligand and receptor interact. And then this would allow us to be able to make use of this type of intuition and knowledge of the structures for being able to approximate the binding affinity and the mode of interaction. In the main paper reference, I'm going to be describing and how we can make use of different aspects of this paper from the aspects that I discussed in another presentation on the channel on protease inhibition, is that we can look at unveiling the molecular mechanism of SARS-CoV-2 main protease inhibition from 92 crystal structures. So this group of authors was looking at 92 available possible crystal structures for the COVID receptor, and how having information on different types of these crystal structures can then allow them to make turn this into a classification type of task, in which we can statistically discriminate between different crystal structures and their primary modes of interaction, and how this would infect, or how this would impact the ability of the coronavirus to infect a host organism. And from this quantitative type of perspective, to gain some type of helpful structural insight on different compounds, we can think about the COVID crystal structure, in which they provide a distribution of the possible binding pockets, there being 12 of them, and we can look at some binding pockets which are aligned amongst the left-hand side of the receptor, and then other ones which we can obtain by doing a counterclockwise rotation to look at the 3 binding pockets on the other side of the receptor. And as I mentioned, this is going to just draw some similarity in a more abbreviated short form to another presentation that I gave on the channel, in which we would like to further demonstrate from a different type of perspective how having a structural biology point of view can help us understand the timescales and different ways in which interactions can be mediated, and possibly how unwanted toxicity can be imparted from mRNA vaccines. But then, but despite the fact that there can be some benefits to such vaccines, we need to actually determine how different conformations could be correlated from this. From that are generated from the machine learning workflow that can help us understand what would be the severity of an infection, or what would be the probability of a vaccine being able to successfully block a certain infection occurring from the conformations that we can study in a structural biology way. And from the first paper reference that we had mentioned on the protease inhibition, this is important because we can look at different types of PDB-IDs, and the PDB-ID is allowing us to look at different types of IDs that are publicly deposited on the protein data bank that have each one of their own characteristic type of structure. And then from the ligand ID, which is given in the next column, we can look at the total different number of bonds or different composition of residues which appear, as well as how different parts of the chemical structure are bonded together, which would then impact some type of structural constraints on the type of conformations that we're looking to try to quantitatively study and approximate through an optimization problem, and how that will then result to some type of classification from a machine learning, or even like from the reference that we've been talking about a deep learning algorithm, or workflow, because this is going to be used to decide or to determine the orientation and the resulting approximation for the binding affinity. And the structural information from the protein data bank is related to the fact that we have a PDB entry, and we have a bunch of information which is following besides this publicly deposited compound, in which it provides the entry authors as well as the deposited date and what would be any revisions, because there could be some types of differences to the chemical structure which happen, and these resulting differences could have implications for the types of conformations within an ensemble of possible states that the machine learning classification or the deep learning classification would settle on, as the algorithm is compiled in some hopefully polynomial time, obviously, so that we can generate many possible conformations for them, comparing in terms of assay development or other experimental procedures, what will we likely be able to see in the lab. And we can explore conformations by sharing connections with the ones below. In that, from these types of conformations below, we can already see how there can exist a rich structure and a rich ensemble of possible structures that can impact the type of approximated binding affinity that we would like to get, and from the types of compounds that we can actually handle from an algorithm with a pretty fast polynomial runtime, so that we can just check out many different types of conformations and states and adjust either different parameters which we feed to the algorithms. In these types of parameters, or in these types of algorithms, a big thing that's important to look at is to choose appropriate binding coordinates, so that the algorithm can determine which region of the surface of the structure of the receptor, which region of some shared surface between even larger scale molecules would likely be able to exhibit some interaction that can be likely captured by some type of assay that can hopefully be iterated on and further developed for more sophisticated measurements of binding affinity. And so this is important because, even like we see from all these types of conformations, there can exist different types of ways depending upon the force field that someone uses, in which the compound in a ligand can interact with a compound, and then how this would be related to the mode and the strength of the interaction, and how as well the time scale during which this interaction could occur could raise implications for how long a viral infection stage takes place for, and what is the strength of the viral infection, which would then have consequences for the type of mRNA vaccine or other types of vaccine candidates or treatments, and how this would be able to related to the type of time scale of the infection or interaction with a disease receptor that would then inform the types of studies and biological processes that we would want to characterize further. And the protocol workflow for gene prediction, which is also related very much to make use of machine learning workflows and pipelines for computational biology is related to the fact that we can look at an unsupervised method, which can help us generate some types of potential structures or candidates, which we can then iterate upon further with the molecular docking workflows. And this protocol workflow is given by the fact that there are initial steps in a main cycle, and basically these types of steps which occur in which initially in the first stage a heuristic model is built and then iterated upon is important because it determines some initial favorable condition within the sequence space that we would like to explore, so that either the algorithm doesn't perform a faulty type of classification and can actually give us a sequence within a reasonable length, which can potentially have some conserved sequences or other desirable properties, and how we can then make use of that information for enzyme discovery in a molecular docking workflow. And the output from the hidden Markov model, which is just another example of an open source machine learning framework, is given by this. And we can look at generating further sequences from a transcriptome assembly, which we'll also describe previously later in the presentation, and how, depending upon the type of information which we feed it, it can generate some genes which can have pretty variable length. And that's why it's important for us to think about in our own type of proprietary algorithms and neural networks and other related structures for computation, how this would actually determine how we can tailor the output of an algorithm so that we can either have the most desirable conformations or the most desirable or from a good classifier that are produced from sequences, which we can then study for other assay purposes or other experimental purposes. And from the classification, we can see that in the FASTA output file, which is another type of genomic FASTA, genomic file format, which I discuss on other presentations on the channel, the FASTA output is able to generate different types of genes as well as variable lengths within the left end and the right end. And then from gene lengths ranging from 99 to even a little more than 300 with 327, it can classify or group these genes together depending upon a classification number. And the Trinity transcriptome workflow can even be used also in conjunction with a molecular docking workflow, because we can think about making use of some sequencing source, which results to some RNA extraction, and then feeding the sequencing source through some graph preparation, in which in the chrysalis stage and the second stage, contigs are grouped together into clusters, which are then forming De Bruijn graphs. And in the third stage, there's a step in which there's a transcript assembly into a multi-FASTA file. And then with the T-BLAST, we can isolate potential gene candidates from generated transcripts, which can inform assay development for further experimental investigation, like how we had been alluding to previously. And the banded graph assembly allows us to make use of some visual type of representation for determining the probability that we actually obtain a correct readout for a gene that we can further experimentally investigate for determining if there are certain properties and characteristics of the sequence from what we know from either a literature source or what we could expect from experiments on closely related sequences in the genome. And other bioinformatic pipelines, including Augustus, relate to prediction of sequences of the transcriptome or genome, in which we can make use of for processing steps for Augustus, a hidden Markov model, which is a very universal type of model in which we look at the probability of different nodes within a network interacting with one another. And we can make use of this for looking at how we can fashion machine learning output from which we can perform as given in the third step, a base-ed local alignment search tool that helps predict genes to form gene pools for future integration and testing, in which we can call this as a type of nickname, a local search pool. But despite potential benefits, vaccine candidates can impart undesirable perturbations on the genome or other systems within a host organism, in which this presentation of one of many is talking about how mRNA vaccine therapy is potentially a hypothesis because it can carry many potential health risks, which can even serve as to parallel potential health risks and undesirable outcomes that can occur from gene editing with CRISPR-Cas enzymes in synthetic biology. And to do this, I just wanted to demonstrate some aspects of the data that was gathered in a synthetic biology system from a colleague of mine, David Speck, in which it was looking at a workflow for measuring DCAS fitness change after binding, where the fitness change is resulting in some interruption to reverse transcription of DNA, which can have some type of larger scale impacts for different parts of us of biological systems that can parallel and share some similarities with the potential health benefits that can result from mRNA vaccine candidates. And in this case, even for the measuring the dead Cas or catalytically inactive Cas nuclease change, we're choosing a viral genome of interest, whether it be SARS coronavirus, influenza, or Zika. And after performing some genome mining in a way that we had illustrated in algorithms previously in this presentation, we can look at generating possible sequence from either transcriptome assembly or genome assembly, as well as fashioning or constructing training set data for each one of these algorithms, which would relate to sequencing resources from RNA. And in this case, if we want to either target an RNA target or even a DNA target with some CRISPR-Cas nuclease of interest, we can measure the DCAS or the catalytically inactive nuclease binding activity through next generation sequencing. And in the second step of David's workflow, we identify editing target sites for dead Cas nucleases depending upon PAM proximal or PAM distal placements, which allows us to obtain hundreds of thousands of testable constructs. And we make use of this workflow because in the same way that we discussed in molecular docking workflows in which there can exist some thermodynamic interaction, which results in a conformation produced from a classification of a machine learning or deep learning algorithm, and how there's a certain time scale that's associated with this interaction between this ligand and receptor, or maybe even larger compounds with each other. For CRISPR-Cas nucleases and gene editing, when we target either DNA or RNA targets, we can look at various types of CRISPR-Cas nucleases, whether Cas9, Cas12a, Cas13a, or Cas13b, which either make single-strand ssRNA or double-stranded DNA breaks. And then each one of these nucleases can be used to target different types of targets within the genome, depending upon whether there needs to be a presence of a tracer RNA, which is like the PAM sequence, because it's another component that's associated with each site that a CRISPR-Cas nuclease may have to target. And then we can also look at the PAM sequence depending upon whether it's G-rich or T-rich and whether it's facing with the 5' end or the 3' end. And with a type of numerical model a couple of years ago, a while ago, we looked at how the solution approximations to some partial differential equation, which can be used to approximate a landscape, the exploration of a landscape potential, can be approximated numerically with this function, which is the exponential of an error function, and how we can tune the parameters over a certain subsequence of the entire basis that's explored either under the DNA or the RNA strand by CRISPR-Cas nuclease to be approximately the order of 200 microseconds, which experimentally agrees with the results from FN-Cas12a complexes. But we can have degradation to components of biological system like we had mentioned, which is just giving another example of a parallel in the difficulty of assay development for testing the efficacy of mRNA vaccine candidates. So besides the fact that there's this production of protease and the protease inhibition is determining how there are mechanisms in which how HIV and COVID can similarly infect host organisms. There's also even a lot of other things like how we have been alluding to in this presentation, and that we can either look to measure RNA degradation as given in the third step of David's workflow through small genetic circuits which are contained in small segments of DNA and plasmids, but the several ingredients from synthetic biology that come into play in comparison to how maybe a virus infects its organism through an mRNA vaccine candidate can be completely different, because we can also expect, despite the fact that there is some differences in the way in which a Cas13a CRISPR system would be able to target some genetic material in comparison to how the RNA degradation could be, or degradation from an infection could be impacted by some virus like coronavirus or Zika. we can still make use of different types of computational workflows to roughly approximate the timescale interactions by which these interactions would occur, which we can then study further with, as we had mentioned, either different types of assays or experimental measures for approximating binding affinity with some narrow set of compounds that we can examine. And toxicity to RNA can be impaired under other circumstances, which can impact other functions of the genome. And this is just another reference amongst many in which the authors discuss RNA toxicity, which can occur in non-coding repeat expansion disorders. And structural information of how receptors and ligands interact can be studied from individual conformations produced in the output PDB-QT file of open source machine learning package AutoDocVina, in which to perform a molecular docking in silico simulation or virtual experiment, we can specify molecular coordinates like I did from the receptor and the ligand, which are each provided in PDB-QT output format, and then we can specify the center of the binding region, as well as the size of the individual coordinates of the binding frame, which would then result in some output PDB-QT format or file. And then we can interpret the results from conformations in the PDB-QT output, in which, like we had mentioned, from the deep learning reference, which provided further details on a deep learning algorithm that was used to study the structure and the surface of the COVID-19 receptor from the crystal structure, we can expect different types of interactions to occur as we perform some either clockwise or counterclockwise rotation of the receptor, and how our algorithms can be used to test each one of these different pockets in which an interaction can occur when we pass different binding frame coordinates to the algorithm. And the implementation steps also include that we can produce each possible conformation, in which, like given in these examples of running AutoDocVina, we can either have 9 or 6 possible conformations produced, each which have a corresponding binding affinity. And we can even see, as we talked about in CellTalk-DB sequences, that there can even be a broader range of disorders or either disease receptors or disease receptors which are associated with different types of disorders or impairments that we can include and look at further for testing virtually at first. For them, we can further discuss in terms of determining which assays would be appropriate for measuring different types of biological phenomena or activity from biological systems. And the CellTalk-DB receptors give a great repository because we can make use of these sequences and this information that's publicly deposited for generating many different types of homology models. And the CellTalk-DB website is in this format, in which they provide different ligand receptor pairs, which we can then search for generating information such as the homology models exhibited in the previous slide. And we can orient ourselves with AutoDocVina because they have an open source software and open source package, which can be used to visualize the docking region and the atomic coordinates that we set amongst each compound and ligand and receptor that we want to study further. But as mentioned earlier in another presentation on the channel, we make use of pre-processing steps for the in-silico approach for converting PDB to PDB-QT files with the Python molecule viewer, which is a pretty standard routine because we either have to make use of some different types of conversion steps with some automated open source Python scripts, which are provided by the developers at AutoDocVina, if we want to feed the crystal structure and the structural information from compounds and ligands and receptors that we want to study and look at into other algorithms for comparing the performance amongst different algorithms. And we can revisit the COVID crystal structure across all inhibitors because it provides different types of information about countably many interactions and countably many conformations, which can be produced from the deep learning and machine learning algorithms for some crystal structures of interest. And we can obtain the PDB-QT files, as we had mentioned, from the protein data bank, because we can also look at different types of biological assembly for the crystal structure of SARS-CoV-2. And the concluding remarks is that we introduced components from previous presentations on the channel for in-silico workflows in which hundreds of compounds can be screened against each other. And we highlighted the wide variety of biological processes that can occur when the receptors and ligands interact, which can share connections with interruption to reverse replication in systems from synthetic biology. And we demonstrated many areas of current interest for applying machine learning and even deep learning pipelines, whether open source or proprietary, for exploring hundreds of new compounds within enzyme space. And thanks for watching.